morning, everybody. Today I'm going to talk to you about dynamic strain measurement in soft tissues. For a brief overview, I'm going to start off with some general comments about the role of experimental measurement in combination with modelling. Then I'm going to focus more specifically on cardiac strain measurement and current techniques that are deployed clinically, and then talk about the aims and objectives of this specific study. I'll give you some background in the methods that we're using in terms of stereo-optical techniques and digital image correlation, with some coverage of accuracy and the experimental measurements we've been taking, and then conclude with results and conclusions. So this work within our group has been going on for some time now, and an initial motivation for this was driven by inherent variability that's observed during stent expansion. So if we expand the stent experimentally, we would never expect to get exactly the same outcome. Whereas if we typically take a numerical simulation, certainly at the time when this work started, they would be completely deterministic. And an element of this is due to the complexity of the simulation, but there's also uncertainties which generally wouldn't be included in a model that can only be measured experimentally. So this drove the need for dynamic high-resolution 3D characterization of stent geometry. And within that project, we started to look at extending that dynamic characterization to the interaction between the stent and the vessel wall. And that's led us to look at other soft tissue applications. And that's what I'm going to focus on this morning. So I'm going to talk about cardiac strain measurement. This is used to provide an indication of cardiac function. There are applications in diagnosis and monitoring, particularly in patients suffering from heart failure and heart disease. The clinical modalities that are used include ultrasound and tagged MR modalities, and these have been developed because, of course, they can be applied directly within the clinic. However, there still remain significant assumptions which are inherent in the image processing that goes on with these techniques, and that can result in potential artifacts, particularly associated with out-of-plane displacements, because in general, these methodologies in clinical practice are still using planar data. So the aims and objectives of our work was to obtain accurate surface strain measurements during transient large deformations of cardiac tissue. I'm quite precise there because Enrico will talk about some other applications later that are within a very different domain in terms of the size of the deformation. What we're interested in is characterizing the accuracy and precision of the methods that we use in detail, and then extending that to compare with the current clinical methodologies. And of course, to do that, typically, we're not using in vivo measurements, we're coming into the lab. This has been funded through a Marie Curie network VPH case. The website is up on the screen there. I'd strongly recommend going to the webpage. There's a lot more detail about the network. In particular, the network has around 10 academic partners and around 4 industry core partners. We're working closely in this work with the Technical University in Eindhoven and LifeTech Group, who are an SME. The fundamental processes we use are stereo-optics. So if we have a 3D domain and we have specific points on that 3D domain, if we image those with a single camera, we obtain 2D coordinates within that single camera view. If we then apply a second camera to the same thing, we obtain a separate set of 2D coordinates. If we then calibrate to high accuracy the stereo camera system using a control object and combine that with the 2D data, we can actually retrieve the full 3D data. And this is not new, it's been used extensively, particularly in gait analysis. The work that Claudia Matza does is fundamentally based on this technique. If we combine that with digital image correlation, what that allows us to do is not only track geometry, but also track deformation. So if we have an undeformed object and a deformed object, and we image both of those, DIC relies on local variations in image intensity to track displacement. So what we need to do to allow us to apply the algorithms is to apply a speckle pattern. So you can see an example of that on the undeformed image. That will then deform along with the object. And if we image that again, by looking at the position of an original point within the image and using the intensity detail to track the change in that position, we can obtain the displacement of the point. But the particular advantage of this technique is that it's full field. So it gives us 2 displacement components across the entire image at very high resolution. If we combine stereo optics and DIC, we can then do that in 3D. So we can track both the structure changes in 3D and the deformation. This is distinct from 3D scanning, which doesn't allow us to do that. It only allows us to iteratively reconstruct the geometry. We want to look at geometry and strain accuracy. To look at geometry, we first took a porcine heart in the lab and we reconstructed the surface using stereo DIC. This shows the region of interest of the left ventricle. We then 3D print that geometry to create an idealized phantom with exactly the same geometry as our original reconstruction. And then we measure that again using the same DIC method. To do that, we have to apply the speckle pattern to the 3D printed object. And the difference in the geometries gives us an error metric of geometrical reconstruction. To look at strain, we again look at the surface of the porcine heart, and we apply a rigid body motion experimentally. So we actually take the geometry and displace it. With a stereo system, it's not trivial to artificially apply a rigid body motion, particularly in this case where we're not using volumetric data. If we then compute the strain distribution from those translations, this gives us a metric of the error in the strain, because of course from a rigid body motion we expect zero strain. That's the assessment of the accuracy of the method. Now we've started to apply that within real dynamic experiments. This is working closely with LifeTech Group, who have developed this biosimulator platform. What that allows us to do is have a passive dynamic model of cardiac function. Clearly this is very idealized, there's no contraction of the heart, the tissue is harvested and it's fresh tissue, but it's driven by a hemodynamic pump. But this allows us to look at repeatability over multiple cardiac cycles and multiple cardiac samples, which is very good within the system and not so much within other ex vivo heart models. We can also then look at the strain variability that the method reports under changes in hemodynamic conditions, and the system allows us to also alter parameters such as systemic resistance and aortic pressure. So now for some results. In terms of the geometry accuracy, the comparison of the reconstructed geometry shows good agreement for a large region of the region of interest. We tend to get errors at the edges of the region of interest, which is a common observation with DIC methods. So over most of the region, we have less than 0.5 millimeter accuracy, which, to provide some context, is less than 1% of the field of view of this image pair. In terms of the strain artifacts, what we see is that we get consistent results over the multiple translations, and this reassures us that there aren't hotspots within our imaging region where we would expect to see a large strain artifact. And also, the mean strain error is less than 1% for a particular subset size. This is illustrated in the two images on the left. This is the distribution of the strain error over the region of interest. Again, we would expect that to be 0. In this case, we have peak errors of the region of 1%. There's a lot of literature in DIC on the relationship between the correlation parameters and the error metrics that you get. It is possible to reduce that error by changing the subset size, but essentially that compromises the spatial resolution. So I think similar to some of Enrico's work, we find that there is a trade-off between how accurately you wish to measure and the amount of data or the resolution with which you wish to report that. In terms of strain variation in the cardiac biosimulator platform, we've very recently obtained these results, but what we see is very good repeatability across cardiac cycles. So what we're able to do is perhaps use one cardiac cycle to assess function using an ultrasound method, and then another cardiac cycle to use the optical method, because it is challenging to apply them both at the same time. We see some deviation in the strain across the region of interest. There are significant outliers at the edge of the region of interest, which again is due to the DIC method. And there's also perhaps some noise in these measurements due to the spatial resolution, which again relates to some of the results that we found in this technique, and Enrico has also found with volumetric techniques. What the technique shows us is that we are able to very clearly differentiate levels of strain under different hemodynamic conditions. So the plot on the right shows the condition in red is perhaps representative of a healthy porcine heart. So we see strains of the order of 10% over the cardiac cycle. The condition in the blue is much reduced, which might represent a model of heart failure. And we are able to successfully resolve those, and our 1% uncertainty in the strain will not compromise comparison of those conditions. So in conclusion, what we've been able to do up until now is demonstrate that we can make repeatable measurements of high-resolution strain fields in a very dynamic system under large deformations. The next work that we're going to do is to compare that directly with clinical modalities, ultrasound, spectral tracking, echocardiography, and also potentially within this project extend it to look at the LiveTech ex vivo beating heart platform, where they're able to take a heart out of the pig and animate that using electrical stimulus. I'd like to finish by acknowledging that almost all of this work has been done by my PhD student Paolo. He has a poster, it's number 22 in the exhibition, and also acknowledge the contribution of our collaborators and the funding from VPH case. Thank you.
Thank you, Andy. Was that a video of bread rising that you showed?
So the example in the lab was actually an elastic membrane, just as a toy example. So we have a left and right image of the elastic membrane and then the inflating membrane.
I look like dough in a bit. Anyway, we've got time for one or two technical questions, more technical questions.
Andrew, are your errors related to the curvature of the surface?
No, so the curvature of the surface should be captured. Essentially, that depends on the resolution that we use for the DIC process. But I think the issue at the edges of the region of interest, there is some evidence in the literature that the shape function that's used within the DIC has a very strong effect on capturing the curvature at those edges, and that's why we might expect an artifact there.
So are these anaesthetised pigs or...
So I should have made it very, very clear. The work that's done in Eindhoven is in collaboration with the slaughterhouse. So actually, all of the material that's used for these experiments is material that would have been used for human consumption and has then been retrieved for use in these experiments. So there's no need for additional animal sacrifice.
It's just there are places in the UK where they do sort of advanced trauma support, for example, at Stoke-on-Trent, where they actually have anaesthetised pigs, and they're doing, these are surgeons practising their resuscitation techniques. So there may be an opportunity there if you against it.
Now that would be very interesting. There are a couple of reports now where people have attempted to use this technique in the clinic. So open heart surgery is one example, but I would say that that's very early research. But having access to those kind of platforms would be excellent.
Okay, let's thank Andy again.